Artificial sweeteners are those seemingly harmless ingredients often found in diet sodas, sugar-free snacks, and even chewing gum. But what are they, and why are they so widely used? Artificial sweeteners are synthetic sugar substitutes designed to provide sweetness without the calories of sugar. They are often marketed as a healthier alternative to sugar, helping people manage weight and diabetes. But recent research suggests that these sweeteners may come with hidden dangers. Names like aspartame, sucralose, saccharin, and acesulfame-potassium. Each has a different chemical structure and sweetness intensity. From diet sodas to tabletop sweeteners, they are ubiquitous. This widespread use normalizes their consumption. A groundbreaking study conducted by Touvier et al. And published in the British Medical Journal in 2022 shed light on the potential link between artificial sweeteners and cardiovascular diseases. The study, involving over 100,000 French adults, found a significant association between higher artificial sweetener consumption and an increased risk of cardiovascular events, including heart attacks and strokes. Participants consuming the highest amounts of artificial sweeteners had a 9% higher risk of cardiovascular conditions like stroke and coronary heart disease. Aspartame and acesulfame-K in particular were linked to this elevated risk. This finding raised serious concerns about the long-term health effects of these seemingly innocuous sugar substitutes. The suspected cardiovascular harm could stem from several biological processes. Artificial sweeteners, especially aspartame, may interfere with nitric oxide production, a vital molecule responsible for maintaining the flexibility and dilation of blood vessels. Reduced nitric oxide levels can impair endothelial function, leading to higher blood pressure and increased arterial stiffness, which are key contributors to cardiovascular disease. Additionally, artificial sweeteners are believed to promote chronic low-grade inflammation. Chronic inflammation damages blood vessels and accelerates atherosclerosis, the buildup of plaque in the arteries. Furthermore, animal studies have shown that sweeteners like acesulfame-K can increase oxidative stress, a condition where free radicals overwhelm the body's antioxidant defenses, further damaging cells and tissues involved in maintaining cardiovascular health. After adjusting for potential confounding factors, the researchers observed a clear correlation between artificial sweetener consumption and cardiovascular risk. While the study did not establish a definitive cause and effect relationship, the observed association was strong enough to warrant further investigation. The results challenged the previous notion that artificial sweeteners are a harmless alternative to sugar. A pivotal study by Suez et al. In 2014 revealed the disruptive effects of artificial sweeteners on gut microbiota and glucose intolerance. The study showed that saccharin, sucralose, and aspartame induced glucose intolerance in both mice and humans. Even a short-term consumption of saccharin caused dysbiosis, an imbalance of gut bacteria, and impaired glucose regulation in humans within 1 week. These changes disrupted the gut ecosystem, impairing blood sugar regulation. The study highlighted the potential metabolic dysfunction caused by artificial sweeteners. Artificial sweeteners can disrupt gut health through various mechanisms, including alterations in bacteria composition and increased gut permeability. These sweeteners can selectively promote the growth of certain bacterial species while inhibiting others, leading to huge imbalances in the gut microbiota. One proposed mechanism involves the fermentation of carbohydrates by gut bacteria. Typically, beneficial bacteria produce short-chain fatty acids that help regulate metabolism and reduce inflammation. Artificial sweeteners disrupt this process, potentially leading to increased gut permeability, a condition known as leaky gut. When the gut lining becomes more permeable, it allows harmful substances like lipopolysaccharides, LPS, to enter the bloodstream, promoting systemic inflammation, which is a key driver in the development of metabolic disorders. These imbalances can disrupt the gut's ability to perform its essential functions, including nutrient absorption, immune regulation, and protection against pathogens. Saccharin, for example, appears to increase the abundance of bacteria associated with negative metabolic outcomes, such as those that promote fat storage or inflammation. The altered microbiota composition affects the body's ability to metabolize glucose properly, potentially triggering insulin resistance and glucose intolerance, which are precursors to metabolic diseases like type 2 diabetes. The disruption of gut microbiota and increased gut permeability can have cascading effects on overall health. A comprehensive study conducted by Debras et al. And published in PLOS Medicine in 2022 investigated the potential link between artificial sweetener consumption and cancer risk. The study, also involving over 100,000 French adults, found a positive association between artificial sweetener intake and overall cancer risk. This finding raised concerns about the potential carcinogenicity of these widely used sugar substitutes. After adjusting for potential confounding factors, the researchers observed a statistically significant increase in cancer risk among individuals with higher artificial sweetener intake. This association was particularly pronounced for certain types of cancer, such as breast cancer and obesity-related cancers. While the study did not establish a definitive cause and effect relationship, the observed association was strong enough to warrant further investigation. The results challenged the perception of artificial sweeteners as harmless alternatives to sugar. Several mechanisms have been proposed to explain the potential link between artificial sweeteners and cancer risk. One potential mechanism involves the breakdown products of aspartame, a commonly used artificial sweetener. Aspartame is metabolized in the body into methanol, formaldehyde, and diketopiperazine. Formaldehyde is a known carcinogen, and some studies suggest that exposure to even small amounts of formaldehyde may increase cancer risk. Chronic exposure to formaldehyde and methanol through the ingestion of aspartame could potentially contribute to DNA damage, one of the early steps in carcinogenesis, the formation of cancer. Moreover, some studies suggest that formaldehyde can induce mutations in genes that regulate cell growth and repair, further elevating cancer risk. Artificial sweeteners also increase oxidative stress, which leads to the production of free radicals. Free radicals are highly reactive molecules that can damage cells and tissues, including DNA. Over time, this oxidative damage may lead to the initiation and progression of cancerous growth, particularly in tissues exposed to high levels of sweeteners, such as the gastrointestinal system. Given the potential health risks associated with artificial sweeteners, it is advisable to minimize or avoid their consumption. Here are some practical tips for reducing your intake of artificial sweeteners. Read food labels carefully. Check the ingredient list of packaged foods and beverages for artificial sweeteners. Choose naturally sweetened products. To avoid the risks, check food labels for common artificial sweeteners like aspartame, saccharin, and sucralose. These are often hidden in processed foods, diet beverages, and sugar-free products. Opt for products sweetened with natural sweeteners like honey, maple syrup, or dates. Limit consumption of diet sodas and other artificially sweetened beverages. Be mindful of sugar-free or low-calorie options. Cook at home more often to control the ingredients in your meals and avoid hidden sources of artificial sweeteners. The evidence suggests that artificial sweeteners may not be the harmless sugar substitutes they are often perceived to be. From cardiovascular concerns and gut microbiota disruption to potential cancer risks, the hidden dangers of these artificial sweeteners warrant careful consideration. By making informed choices and opting for healthier alternatives, we can prioritize our long-term health and well-being. The allure of calorie-free sweetness can be tempting, but the potential health consequences associated with artificial sweeteners should not be ignored. Embracing natural sweetness through fruits, vegetables, and natural sweeteners like stevia and monk fruit, we can avoid the hidden risks of artificial sweeteners and consume foods that can benefit overall health. By focusing on whole, unprocessed foods and minimizing our intake of artificial sweeteners, we can cultivate a healthier relationship with sweetness. Please like and subscribe for more nutrition guidance. Stay healthy.